Hello and welcome to XploreBio. You all must be aware of novel coronaviruses, and now people are more frequently talking about their variants or mutants. So today we will learn what are novel coronavirus or SARS-CoV-2 variants, where they came from, are the variants more deadly, are the existing vaccines effective against these variants, do RT PCR and rapid antigen tests work well for detecting these new variants. SARS-CoV-2 was initially reported in December 2019. Since then, it has infected millions and millions of people across the globe. At one hand, where governments and scientists have found ways to fight against it and reduce its transmission rate, the virus has been proved much cleverer than we think. Every effort, whether it's lockdown, social distancing, using masks and sanitizer, seems not enough to stop this virus. The virus is continuously mutating and evolving. But why is it happening? To know this, we need to see how the virus infects and transmits in its host. SARS-CoV-2 binds to the host human cells through its spike protein at its surface. Upon binding, it releases its genetic material, which is RNA, into the human cell. The virus quickly multiplies its genetic material using host replication machinery. During replication, there are chances of mistakes which leads to change in nucleotide or mutation in the genetic material of the virus. A SARS-CoV-2 variant may have one or more such mutations. Randomized sequencing of SARS-CoV-2 at various parts of the world has resulted in identification of more such novel variants. There are several variants of SARS-CoV-2 denoted by Greek letters. Alpha, beta, delta, gamma are the most commonly reported variants. Alpha was first detected in UK, beta in South Africa, gamma in Brazil, and delta in India. As the mutations are random, there are several variants that have low transmissibility and are less infectious, which are of less concern, while others although have high transmissibility but poorly infectious, while few shows high transmissibility and more severe disease symptoms. These are the variants of concern. For example, the Delta variant that has high transmissibility has led to the second COVID wave in India. Also, it has led to more hospitalization cases than earlier strain. It took almost a year and a half we have vaccines for novel coronavirus. We have a variety of vaccines for the virus, including inactivated virus vaccines such as Covaxin. Then there are viral vector vaccines like Covishield, Johnson & Johnson's, and Sputnik's. And then there are mRNA vaccine from Pfizer and Moderna. All the governments have tried their best to vaccinate its people, but the big question is whether the vaccines be effective against the novel variants. One thing is clear that all the vaccines that have been developed were built to provide immunity against the original strain of novel coronavirus and not its mutant. Based on the existing information, it is clear that the vaccines are good for the original strains, but several reports have shown marked reduction in vaccines efficacy up to several folds for newer strains. And no vaccine is 100% effective against all the variants. It will become clear only with time upon analyzing populations which are vaccinated and disease severity they show. But whatever the case may be, the vaccinated people will have at least some advantage over the unvaccinated people. Now the question comes if the variants can be detected by RT PCR and rapid antigen test. The RT PCR targets a specific gene of novel coronavirus. If the target region where the primer binds is not mutated, the test will work fine. Similarly, with rapid antigen test, which targets a specific epitope of the spike protein, if the epitope does not get mutated or only gets slightly modified, the antibody will bind to it and the kit will produce correct result. In general, the diagnostic kit should work well with the variants too. Still, due to technical errors and low viral load, sometimes the kit will produce false results. In such cases, the test needs to be redone. For better diagnosis, the symptoms of the patient should also be considered. So that's all for today. If you like the video, do share with others. Mention in the comments about the videos you might be interested. Check my other interesting videos related to coronaviruses, research publications, techniques and lot more. Subscribe and hit the bell icon to get notified about my latest uploads. And finally, thanks for watching.